RER, rough endoplasmic reticulum, has a histological nickname. It's called Nissl bodies. And that's one way that you can stain for cell bodies. One way to highlight a neuron cell body is to stain the RER because it's so extensive. It kind of highlights the whole shape of the cell body itself. Like right here. This is not a stain that is meant to highlight the cell body as a whole or in general. It's actually just to stain ribosomes. But in doing so, you've basically highlighted the whole cell body. There are so many ribosomes. Fixed ribosomes, RER, and free ribosomes as well. What do you think AH is right here for this triangular structure? Axon hillock, yep. And then the axon coming off of it. Neurofilaments, which include neurofibrils, form the cytoskeleton. So the second image, all of these from the histoatlas. Here they actually stained neurofilaments. It's all these dark, kind of dark orange, brown lines. And they're in the cell body and they're in the axon. They form the framework of the entire neuron. The membrane, and we're talking about cell bodies still. The membrane acts as a receptive area for receiving signals, which means, what does it have in the cell membrane?
Receptors.
Neurotransmitter receptors. Terminology. Cluster of cell bodies in the PNS is called ganglion, singular, ganglia, plural. And here's the confusing one. A cluster of cell bodies in the CNS, brain or spinal cord, is called a nucleus, which has nothing to do with nucleus of a cell. In a more general sense of the term nucleus, it's a localized cluster of something. Cluster of cell bodies in the brain and spinal cord. So in chapter 12, when we start looking at gray matter, we're going to have lots of named nuclei. The thalamus, for example, which is all gray matter, can be divided up into about a dozen different nuclei. Each one has a name. And of course, what we're referring to in that case are groups of cell bodies. Neurons grouped together often have a common function. They're all working together to process similar information. Processes. Neuron processes extend from the cell body. There's two types, dendrites and axon. Dendrites have neurotransmitter receptors. It's the main receptive area of the cell. Branching increases the surface area and can include up to 90% of a neuron's total surface area. I'll give you an example of what that can look like. Far left. This is a particular type of neuron called a Purkinje cell. It's found in the cerebellum. Here's the cell body. Notice there's one axon and two dendrites that come off of it, but then look at how much the two dendrites branch. And you can call the branches dendrites as well. Dendrite is just a generic term for a spine, a process, a branch in this case. But I usually start at the cell body. Whatever comes off of the cell body, that's how I number the dendrites. And then if they branch, then those are dendrite branches. So out of all these cells that are shown, you can easily see which one receives input from the largest number of neurons. The one on the far left has the biggest receptive area. Dendrites conduct signals, and the direction is towards the cell body. And those are local potentials, graded potentials. As they spread to and through the cell body, they have to reach the axon hillock. And when they reach the axon hillock, those graded potentials have to be strong enough to bring the membrane to threshold. So rest is maybe minus 70 millivolts. Threshold, let's say, is minus 55. So that means you have to have a 15 millivolt current, a positive current, the strength 15 millivolts, upon reaching the axon hillock to bring the membrane to threshold. If you don't bring the membrane to threshold, no action potential. And then what happens to that graded potential? It just decays. Nothing happens. So they vary in size, and they're supposed to, and they don't always result in APs.
So in the homework, local potential would be graded potential doesn't transmit AP?
Say again?
Local potential?
Which is graded potential. Local is graded.
Oh.
So there's only two types of potentials, action and graded, or you could say action and local. But local means graded. And what they mean by the term local is it doesn't spread very far. It doesn't renew itself, it doesn't propagate. Decays over a very short distance. A single action potential decays as well. The difference is, it's going to open another batch of channels and renew itself. So you have one axon. It can be short, it can be really long, it can go from the spinal cord to your big toe, in which case it's several feet in length. Lower motor neurons tend to be quite long, right? They go from the spinal cord to every muscle, no matter how far it is from the spinal cord. Terminology related to the axon, nerve fiber. Nerve fiber is the axon. Collaterals. You can have branches that come up. And these aren't the branches at the very end. And I was hoping I had an example somewhere. There we go. The branches at the end are called telodendria. And that's what these are, and these. But if you just kind of make a right angle right off of the axon, somewhere down the middle of its length, that's a collateral. And what you often see is axons that are communicating with lots of different cells tend to have more collaterals. A branch can go over here, and a branch can go over here, and you can communicate with different cells at the same time. You have the axon hillock, the axon itself, the telodendria, which are the branches at the end of the axon, and then the bulbous endings, which have 5, 6, 7 different names. I usually call them axon terminals. Some other names you might see: synaptic knobs, terminal boutons, end bulbs, neuropods, and there's others. Axon terminal is simple and to the point. Axoplasmic transport. Right, the main job of the axon is carried out by the membrane, the axolemma. And what's the function of the axolemma? Conduct action potential. That's the main job of an axon, conduct action potentials. Inside of the axon is the inside of the cell. And you often need to transport things across the length of the cell, from one end to the other. For example, we say that the cell body is the biosynthetic center of the cell, where you have the nucleus and most of your ribosomes. And so that's where you make proteins. But if those proteins are neurotransmitters, you don't need them in the cell body. You need them to go somewhere else. All the way down to the very end of the axon, to the axon terminals, where they can be stored. So how do you get them there? Axoplasmic transport. Right? And you can find some animations online. Just Google kinesins, axoplasmic transport proteins, and they'll show you these little animations where these little motor proteins literally walk down the microtubules. It's like a conveyor belt. And there's two variations of axoplasmic transport, slow and fast. And the fast is quite fast. And it's a way to shuttle materials back and forth. So at the same time, you may want to bring neurotransmitters from the body to the axon terminal, which is forward transport, anterograde. You often pick things up in the axon terminal, or somewhere along the length of the axon, then you want to take to the soma, the cell body. Especially chemical signals, things that are regulating genes and gene activation. Unfortunately, there are some bacteria, some viruses, that have figured out how to manipulate axoplasmic transport to bypass the blood-brain barrier. If the axon is in the peripheral nervous system, in which case it's exposed to blood, and say the cell body is in the central nervous system, either the brain or the spinal cord, maybe it's a sensory neuron, could be motor, doesn't matter. The virus can sneak into the axon terminal, hitch a ride, retrograde, backwards, retrograde axoplasmic transport, reaching the cell body. Now you're inside the brain, the spinal cord, the CNS tissue, and then you can get through the cell membrane and basically infect the nervous tissue of the brain or spinal cord. Polio virus, rabies virus, toxins, some bacteria bypass the blood, the ECF, the blood-borne immune system, and travel through the axon to nervous tissue, and thereby also bypassing the blood-brain barrier.
That can only happen in retrograde, never in anterograde.
Correct. Now, in some of the research I did, we actually took advantage of retrograde transport and had indicators, basically dye, that the axon terminals would take up and transport back to the cell bodies, so we could actually see where the cell bodies were for certain types of neurons. And that's one way to map out the brain and spinal cord, to figure out where certain types of information goes. So if you can identify nociceptors, pain fibers, and then you can treat them with a dye, you can figure out where the cell bodies are, the nuclei that process that pain information. It's one of many methods that have been developed to map out nervous tissue, figure out where information goes. Primary function of axon, we talked about that. Schwann cells forming myelin. talked about that. Oh, I pushed the end button, didn't I? I hate it when I do that.
So? I was wondering where you were.
I try to push down and I hit end on accident. It skips to the very end. The only other thing I have regarding Schwann cells, a couple of depictions of how myelins actually form. So here's the initial Schwann cell. Here it is as it begins to wrap around. Those inner layers, they get wrapped really tight, tighter than what they're showing here, becomes the myelin sheath. And then the outermost layer, which has a new name. Old name is neurolemma. New name is perinuclear cytoplasm. That's where the cell body is of the Schwann cell. So neither the myelin is depicted correctly, nor is the neurolemma. Neither is depicted correctly there. So here's the real thing, and you'll see what I mean. Here's the myelin. That's how tightly wound those layers are. It looks like a solid black band. But if you could look really, really closely, there's dozens of little lines there where each layer is wrapping again and again.
It's in the bigger one.
You can see it over here. Exactly. That's how tightly wrapped the cell membrane is to form the myelin. And then way out here, that's how big the nucleus is. Just the nucleus of the neurolemma. And then this is all the cytoplasm out there around it. So those cartoon versions are not realistic at all.
So it's supposed to be like a roll of tape.
Oh yeah. Yes. Very much like a roll of tape. In these bottom two images, they used a stain that gets picked up by lipids. It stains lipids. And by staining lipids, you essentially stain the myelin, because that's what myelin is. Lipid bilayer after lipid bilayer wrapped around and around. So that allows you to clearly see the internodes, which become black, and then the nodes of Ranvier, which lack the myelin and become clear or white. So what's this a cross-section through? Looks like a bunch of tires. What are we cutting through in cross-section?
Through the axon?
Myelinated axons. Lots and lots of myelinated axons. It would be white matter if it was in the brain or spinal cord, or just a nerve. Right? A nerve is a bundle of nerve fibers. Take a break. We'll come back to classification of neurons. Take a 10-minute break.